So good morning, everybody. Today we are covering chapter 10, genetics. We will be doing some Punnett squares for this, and this is a long chapter, so we are going to spend 2 lecture periods on this chapter. For this chapter, you need understanding of the previous chapter, which was sexual reproduction, because it's going to come up again in this chapter. If you don't understand meiosis, you might have hard time understanding Punnett squares. So genetics can explain how these kittens look different. Sexual reproduction. I showed you this picture last time as well. And I said that through sexual reproduction, half of the genes come from the mother, half of the genes come from the father. And then the kids, they might have some genes that are the father and some from the mother. So what you see here, they don't look exactly like the mama cat. They look different. That's because some of the genes are coming from the father. So genetics, they started studying it before they knew that DNA is double helix. So studying genetics shows that how to basically they can select for some genes in crops. They can also, when they're breeding dogs and cats, sometimes they select for the genes. So they can select for it, and then they can pick the genes that they like, and then they can do breeding like that. And then if you go to the doctor and you want to know, maybe there's a genetic disease going on and in your generation, you want to know what is the chance for you to have that disease. They can, do some calculations and say you have 25% chance of having maybe whichever disease that you were talking about. So that's what we're going to do today. Have you guys heard about CC the cat? Have you guys heard about CC the cat or Dolly the sheep?
I think so.
Catherine, do you remember anything about CC the cat?
No, I just kind of remember the name.
Okay, so CC the cat was this first cloned cat. They took some skin cells from this cat, and then they cloned another cat. So this cat is a twin of this cat. Basically, they have same genetic information. But then you look at it and you say, oh, this cat looks completely different than this cat. And that's because not the same genes got activated. So when you even when you have the same genetic information, you might still end up looking different because different genes got activated. And it depends on the environment as well. So environment plays a role in activating genes as well. So just because you have the same exact genetic information doesn't mean you're going to look exactly the same. So it depends on which genes get activated. Dolly the sheep was the first cloned sheep. So they did it from skin cells, I think.
Is that the same thing from identical twins and fraternal twins?
It's little bit
Or
different because in this they're taking the skin cells, and for identical twins it's the zygote that's splitting in half, and then in fraternal twins it's getting two zygotes that are getting fertilized.
okay.
So this is a review. I gave you guys few vocabulary words last time. You have to know these in order for me to move on with this chapter, or for you to understand this chapter. So one word was homologous. Homologous chromosomes have the same genes, but they might have different versions, allele. This is very, very important that you understand what allele means for this chapter. Allele is a different version of a gene. Homologous chromosomes are going to be same length, and they're going to have same genes, pretty much on same locations as well. You're going to see this word locus. Locus, this is the physical location of the gene. So locus means physical location, where it's located on this chromosome. If you open up the nucleus, you're gonna have the chromosomes, and this is karyotype. Inside the nucleus, there are the chromosomes. This is the karyotype. This is all the chromosomes that there are for humans we're talking about here. And these chromosomes are going to have the genetic information. Another word I said that there is 23 pairs, and 1 to 22 non-sex chromosomes are autosomes. 23rd one is the sex chromosome. For females, it's XX. For males, it's XY. Um, and then make sure you understand what homologous means. Homologous. So they're same length and they have the genes located at the same location. And the location is known as locus. Locus. This is gene A locus, gene B locus. Um, Another thing is allele. So the best analogy I can use to explain allele, maybe think of it as, so think of this gene maybe as a dessert. So think of this big A ice cream, this cake, big B cake. Now this gene is for ice cream, this gene is for cake. So if you have big A, maybe it's vanilla ice cream. Big A, vanilla ice cream. Here, big B, chocolate cake. If you have little b, maybe vanilla cake. So it's still, the gene is for cake, but it's a different version of that cake. So that's allele. So make sure you understand what allele means. I want to stop here and I want to ask if there are any questions on any of the words or information I just covered on this slide. No? Yes, no? Okay. So another thing is diploid. That's having two copies of the chromosome. So diploid cells have two alleles for each gene. So two alleles for each gene. These alleles might be identical, gene A or different gene B. So they can be same. So for this one, it's the same, big A, big A, big A, big A. Here it's different, big B, little b. So it can be same or different. Each gene's locus is the location on the chromosome where the gene is located. So locus is location. So this is the locus for gene B, this is the locus for gene D, and this one is for A. So that's the location. Physical location of the gene is locus. So usually I want you to know some of the names of the famous people who contributed to science. One of the names I need you guys to know is Gregor Mendel. He's considered the father of genetics. He was the one who was able to figure all this out, and he used pea plants. And he was not even a scientist. Look at his clothes. He was a priest. He was a priest. He was not a scientist, and his hobby was to grow pea plants. And it was nothing new. So agriculture has been happening for long time, and growing pea plants is type of agriculture. So what he did different was when he grew pea plants, he kept a record. He took meticulous notes. So what he did, he would grow all different kinds of pea plants. Maybe this pea plant is tall, this pea plant is short. And then what he did, he would take some pollen from this tall plant and he will brush it onto this smaller plant. And then he would get a hybrid. Then he would plant the hybrid and then he will count how many tall plants he got and then how many short plants he got. There's a show, there was a show called Mythbusters. I don't know if you guys heard about it. So the person on that show said that difference between doing science and goofing around is that you keep a record. So he kept a record. He took very meticulous notes about what he was doing. If he was only doing self-fertilization, means he wasn't mixing anything, or if he was mixing something, and then what was the results? And if he did this again, what would be the results? He even predicted if I mix this plant with this, or if I take some pollen from this plant, brush it onto this, what would be the result? And then he did the Punnett squares. Another thing is he had no information about the DNA at that time. And then Darwin's theory of evolution was happening at the same time. So two of the biggest, important parts of science were happening at the same time, and they didn't know about each other. And maybe because he was not a scientist, he was a priest. And then Darwin's theory of evolution came around the same time. So I need you to know the name Gregor Mendel, and he was the one who got who was the father of genetics and who discovered the basic laws of inheritance. So hand pollinating plants allowed him to control plant breeding experiments. So he just took some pollen from here, brushed on to this plant. So he did hand pollination. Self-fertilizing, that would be just, the same plant getting fertilized. There's no cross-fertilization. In different combinations allowed him to deduce the principles of inheritance, which means he was able to get the purebred. So if you keep doing self-fertilization over and over, you're gonna get a pure breed or pure plant. In dogs and cats, what they do sometimes to get the pure breed, they just they do the inbreeding. They do inbreeding a lot. So if there's a dog or a cat that's been inbred a lot, and then they're not going to live very long, and I'll tell you why that in a little bit. So if you're maybe planning to get a pet, maybe get a mixed breed, not a purebred. So true breeding plants produce offsprings identical to themselves. So this is what I meant when I said purebred, also known as true breeding. So you have this pea plant that gives you green peas. If you keep doing self-fertilization, so take some pollen from this plant and keep brushing onto the same plant, you keep doing this over and over and over, eventually what you're gonna get a plant that's only going to be for the green peas. So self-fertilization is going to give you offspring with the same seed color as the parent plant. Then that means the chance of having yellow seeds has been completely erased, basically. So eventually you're going to get a true breeding plant that only gives green plants. It's only for green peas. Same thing for this yellow pea plant. You do self-fertilization, eventually you're gonna get a true yellow pea plant, true breeding yellow pea plant. Now, because this is only green and this is only yellow, you can decide to cross-fertilize these. So you can take some pollen from this plant, you can brush it onto this plant, and you get a hybrid. So now, because you mix these two, you're going to get a hybrid, and but you don't know how many. So right now here it shows 1 green and 3 yellow. We'll look at why that's the case, why it's not 50-50, or why it's not 1 yellow or 3 green. But anyway, so if you mix this green with this yellow, you get a hybrid. That's a self-fertilization, yields mix of seed colors. So these were true breeding plants. Once they got mixed, you got a hybrid. This is very important. When you're reading the question, you're gonna get, a paragraph. I might say, this is a true breeding plant. You need to keep a note. True breeding, that means it's, one gene. Maybe in this case, it's a gene for this green peas. And in this case, for yellow peas. So when you're reading the question, you need to start underlining things that are important in order to do the Punnett square.